And with that, we'll go ahead and get started. So it's my great pleasure to introduce our speaker today, Dr. Yarelis Gonzalez-Vargas, who is a postdoctoral fellow in the Georgia Clinical and Translational Science Training Program at Georgia Tech and Emory University. And she is working under the mentorship of Dr. Brandon Dixon. She earned her PhD in biomedical engineering in 2024, where her dissertation focused on advanced 3D bioengineered models to study lymphatic malformations. And one interesting fact about Yarelis is last year she was one of the recipients of the Alpha Male Young Investigator Award to study lymphatic malformations. And she's also supported by the Georgia CTSA and the NHLBI Pediatric Clinical Research Program, which bridges tissue engineering, biomaterials, and genomics to develop precision-based strategies for pediatric patients. She's also deeply committed to mentoring future scientists and aims to establish her own independent research program focused on tissue engineered models for pediatric diseases. The title of her presentation is From Biopsy-to-Bench, Engineering Human Lymphatic Tissues for Precision Medicine in Pediatric Vascular Anomalies. Please join me in welcoming Dr. Gonzalez-Vargas.
All
Thank
right. Thank you, Dr. Dallender, and thank you to all the organizers of the CLA webinar for inviting me to speak today. Hello to everyone. Thank you for joining today's webinar. As Dr. Dallender said, my name is Yarelis, and I'm a postdoc fellow at Georgia Tech and Emory University. I work under Dr. Brandon Dixon in the Laboratory of Lymphatic Biology and Bioengineering, and I also co-advised by Dr. Rosanna Sanchez as far as my funding through my postdoctoral training at Georgia Clinical and Translational Science in the TL1 program. So today I want to walk you through my work on engineering human lymphatic tissues from patient biopsies and how this approach supports precision medicine in pediatric vascular anomalies. All right, good to get started. I don't know what happened in my title slide that there's like a drawing, but I did not do that. All right. So lymphatic malformations, or LMs, are congenital vascular anomalies that affect about 1 in every 5,000 newborns. These malformations are typically driven by somatic mutations in PIK3CA, which cause abnormal lymphatic vessel growth. However, because these mutations occur in a mosaic pattern, meaning that only a subset of cells carry this genetic mutation, they can be very hard to detect. And current treatments are limited, which underscores the need for patient-specific disease models that can help us understand how these mutations drive pathology and how we might intervene. So my research pipeline is really built around the idea of bringing patient tissue into the lab, where we start with lymphatic tissue obtained from pediatric patients. These patients are undergoing routine treatments for their lesions at Children's Atlanta. And we expand these tissues in a lymphangiogenetic biomaterial for 30 days. After that, we isolate primary endothelial cells and perform screening and functional studies using an organoid model. We also evaluate how these cells can respond to therapy at the transcriptomic level and map gene expression signatures to uncover mechanisms of endothelial dysfunction and therapeutic sensitivity. To model lymphatic tissue, we use a synthetic PEG formaldehyde polyethylene glycol hydrogel that is highly lymphangogenic, shown here that schematic. This system is modular, where we can control same cell matrix interactions through RGD binding motifs, as shown in here, and control degradability through GPQW peptide crosslinks. So again, this tunability, what allows us is to mimic not only the stiffness of tissues in the lymphatic tissue and the lymphatic malformations, but also modeling that remodeling capacity of real tissue in the body. Importantly, this platform supports the survival and expansion of PROX1-positive cells, or lymphatic endothelial cells, that are isolated via protoplanin or and CD31 magnetic beads, which are crucial for maintaining lymphatic identity. And then we use these to form lymphatic malformation organoids, what I'm showing here, focusing specifically in those lymphatic endothelial cells, because we are interested in the lymphatic endothelial dysfunction. because we do this, we have to validate this platform. So we validate that the cells that we are actually growing in our biomaterials retain their transcriptomic fidelity to the original tissue. We first did this by using single-cell RNA-seq, and I've shown this data before a couple of times where we've shown that in our platform, when we look at the transcriptomics of the single-cell sequencing before expansion in our hydrogel, so the pure biopsy versus the cells that were obtained from the platform, we see that there's not like huge differences between the transcriptomic profiles of these two cell populations, so namely tissue biopsy and the derived cells. And we also see that there's a preservation of key lymphatic markers such as PROX1, which I'm showing here, which confirms again every time. Oh, I keep seeing that drawing. I didn't notice it until now. Is there a way that I can remove that?
I don't know where that
I
is.
don't think that was me. Somehow it just appeared there because I
Yeah,
put a point there. So I don't know. So excuse everyone that there's some drawings in here.
yeah, I don't know what's going on. I'm trying to see if I can clear those, like maybe someone's using a tool.
Yeah, it seems like someone's drawing accidentally.
Okay.
Um, but it's all
I'll,
right.
I'll try to work on this while you present.
Thank you. Yeah, I just checked. I don't think it's me. So anyway, so what we've seen in here is that we are still preserving lymphatic markers even after isolating the cells from a biomaterial and having them in extended culture. So talking about this biomaterial, then I wanted to establish a similar pipeline, but in this case, I wanted to establish this pipeline where I not only expand the human lymphatic malformation tissue from patients in the hydrogels, but we also perform alpelisib treatments longitudinally, shown in here in this schematic of my timeline, and we can compare effective therapy on cell populations after isolating them. After 2 weeks here, what I'm showing is the original tissue, and so in the red cutout and the asterisk in the center shows what the original biopsy segment looked like in the day 1, and then you see at day 14 we see that there is a substantial growth of lymphatic-like structures or sprout all over the around the original tissue, and that happens even when we add alpelisib treatment to the tissue at low concentration. So our goal is not to decrease all of the cell proliferation, but to attack those cells that may have the mutations and see how those that alpelisib treatment longitudinally for 30 days affects the cell populations in our single cell sequencing. Yeah, we also did we used deep sequencing to detect PIK3CA mutations in these cells, and we found that this particular tissue, patient 5, had about 4% PIK3CA mutation in 1047 in the frozen tissue. So we sequenced not only the cells, but also the actual tissue that was frozen right after the biopsy. And then the cells as well after expansion and isolation with magnetic beads for lymphatic endothelial cells. And so we see that even after expansion, we can still retain these lymphatic markers, not only lymphatic markers, but the genetic signature of the disease. However, although our platform is helpful in isolating lymphatic malformation relevant cells that harbor these pathogenic mutations, we've seen that with single cells, we've noticed that there is a high percentage of cells that belong to other cell populations, most likely a stromal origin or any other non-lymphatic endothelial cell related cells. So what I'm showing here is that most of the percentage of our cells in our sample are actually possibly not lymphatic endothelial cells. They are stromal in nature, but however, we still get a substantial amount of lymphatic endothelial cells, which is the purpose of this platform, and we still are able to obtain those mutations from those cells. So when we began this project early on, we initially we wanted to perform whole exome sequencing on the patient-derived cells. This is some data that I've shown before in some of these same groups. And this was done at 50x, and we know that 50x is not enough coverage to identify these somatic mutations. So what this did, it showed us that mostly common germline polymorphism. So that wasn't explanatory for us. And so we then looked at a refined approach, and this was done in collaboration with Dr. Dong Lee at Shaw, where we did targeted deep sequencing. This is done at a coverage of 5,000x, which helped us reveal pathogenic not only PIK3CA mutations, but also we saw mutations in KRAS and HRAS. Here I'm showing one of the examples, the patient 1 with a KRAS mutation, patient 5 with a PIK3CA mutation is the one that I just recently showed. These mutations were confirmed with deep sequencing and validated via Sanger sequencing, which I'm showing here. And we also have one of our patients that showed a 1047 mutation in PIK3CA in the clinic. So this was inpatient genetic testing, not done by us, but we have this cell population as well. And so overall, for those of you to know, the difference between the whole exome sequencing and the targeted is just the coverage of the red. So we just get a lot more coverage in reading those applicants a lot more. And so that allows us to, this high depth approach then allows us to combine the clinical genetic testing, allows us to directly correlate genotype with functional behavior in our models. Now, functionally, we can also observe how patient-derived lymphatic organoids behave in our hydrogels with varying stiffness. From our prior studies, we have shown and know that PEG concentration and therefore matrix stiffness is directly proportional to that stiffness, which strongly influences sprouting behavior. And these assays, sprouting assays specifically, reveal that there's patient-specific variability in lymphangiogenetic potential, which can possibly relate back to their underlying mutations. So here I'm showing those patients that have those KRAS and HRAS mutations identified via deep targeted sequencing. Different levels of PEG concentrations from lower stiffness to higher stiffness. And you can see the sprouting potential differs between patients, especially this patient 1 is highly lymphangiogenic versus patient 2 and 3 are create these just shorter branches. We then also use these models to test therapeutic responses. This is the bulk of our work, just testing therapeutics. So, for example, in here, I'm showing in the PIK3CA 1047 mutant cells, that with treatment with alpelisib, which is a selective PI3K inhibitor, compared differently from sirolimus, which is an mTOR inhibitor, and it's downstream of that PI3K. Therefore, alpelisib being more targeted towards this specific mutation, we showed that there is effect of alpelisib in the lymphatic malformation organoids of this patient that has the 1047 mutation. When we compare it to control lymphatic endothelial cells, non-mutated, we don't see any significant changes in viability, which is expected as these cells are not mutated for the PIK3CA. Therefore, we show that alpelisib is highly targeted towards these mutations. We've also screened these KRAS and HRAS mutant cells derived from LM tissues. And I did this by developing a regression assay to model the dysregulated growth of lymphatics and lymphatic malformations. To do this, I first grew lymphatic malformations as organoids until substantial sprouting had occurred. As I said, this is the untreated version. And then after those sprouts have been grown in the hydrogels, we treated them with alpelisib. And we see that there's changes in the length of branching and amount of sprouts.
I'm going to try, sorry to interrupt, I'm just going to try one thing that I think will clear this. And
go ahead.
Okay. Now, do you mind resharing your screen?
Let me do that. Do I go there?
Perfect.
Okay, there's no more drawings now. Good. So we confirmed this responsiveness to alpelisib by quantifying the sprout regression. And while all patient tissues did respond effectively to treatment with both sirolimus and alpelisib, we found variability in response to alpelisib when compared to sirolimus. Especially here, you can see that this patient did not have a different effect from to alpelisib when compared to sirolimus, but it did have an effect compared to untreated, as you can see in here. We further looked into the transcriptomic response due to regression after alpelisib, so the same experiment, but looking more at the transcriptomic level. To do this, we collected RNA from multiple replicates across these 3 patients, so patients 1 to 3 with the KRAS and HRAS mutations, and confirmed that there was an increase in apoptosis markers and inflammation response markers when compared to an untreated group. In one of our patient models, for example, here I'm showing patient 4, we did single cell sequencing for some of these patients. And so here I show that in patient 4, although patient 4 had no detectable mutation, and there was also no significant transcriptomic response to treatment. So just to remind you, we did this longitudinal study where I was expanding the tissue of the malformations while also adding alpelisib to one of the groups. And so in here, I'm showing that a patient that has no mutation in picture CA, there's no shifts in the populations that show up in the treated versus untreated populations. And by contrast, the patient 5, the 1047R, which was detected via deep sequencing, we showed that it did exhibit changes in cell population and transcriptional programs after alpelisib treatment, which maybe this direct link between genotype and phenotype, it's underscoring again how our patient-derived models can inform our clinical decision-making in the future, because we see a strong response to the alpelisib treatment. Looking a little bit more in depth to this patient 5, we have seen that longitudinal alpelisib treatment of the lymphatic malformation tissues induced population changes that were detected by single-cell sequencing, as I showed in the previous slide. Notably, we saw that tissues treated with alpelisib had a higher recovery of PROX1 positive cells. There were shifts overall in the populations that we saw before integrating these data sets, and then we looked at the data set of only PROX1 positive cells and see that there is a shift of a decrease in PROX1 positive cells in the untreated populations, as shown in the single cell sequencing data. However, these variant allele frequencies in the cells still show recovery of mutant cells in the samples, again, underscoring the capacity of the platform in isolating disease-associated cells for in vitro testing. Now, shifting a little bit, because we wanted to take this platform and insights that we've gotten from all these in vitro studies that we've done, we wanted to move it further by building an integrated platform that combines perfusion control and imaging to continuously monitor lymphatic tissue behavior under flow. This system was developed in collaboration with Lymphatic and Drs. Brendan Dixon and Paco Robles here at Georgia Tech. And this system was made to mimic physiological shear stress and flow conditions while providing label-free imaging through quantitative oblique back illumination microscopy, or QOBM. QOBM let us capture this 3D-like structural information from live samples using reflected light only, which is perfect for dynamic, non-invasive monitoring of lymphatic tissue growth. To the left, what I'm showing there is the video of a lymphatic malformation organoid growing in our platform, and to the right, I am showing how these lymphatic malformation cells in 2D can also respond and align to wall shear stress. To be able to obtain these functional images that I'm showing here in the bottom, a sample is typically imaged over a period of time and its temporal signals are analyzed. Then the dynamic signals can be displayed and in an exponential frequency response that can be analyzed with phasor analysis. And these functional images of the dynamic signatures are obtained by mapping the frequency dynamic response to phasor space and color coding cluster signals shown in here. So I hired dynamic activity of the cells, it's a higher color here, so in red. Then when we pair this QOBN system with time-lapse imaging, our integrated system, or its dynamic version, DQOBN, we can visualize subcellular activity as a functional readout, and that's our goal in this project. For example, we treated cells with alpelisib, lymphatic malformation cells with alpelisib, and we observed there was an increased subcellular dynamic, suggesting that there was an altered metabolic or cytoskeletal activity that corresponds to drug response. This approach that I'm showing here can provide real-time, label-free signature of therapeutic effect, which we have shown in our lymphatic malformation cells, both in 2D culture and 3D culture of organoids.
And I'm sorry, if you go back, I think I missed this. What does the color indicate?
Yes, so the higher color, so the red is a higher dynamic activity, which typically is shown by its subcellular function, so organelle movement or cytoskeletal movement.
Thank you.
So overall, this work has three key implications, as I like to think about it. First is the patient impact. So we can model lymphatic malformations at the individual level to predict drug responses. Second is the scientific innovation where we're providing a human-based platform to study how mosaic mutations drive endothelial dysfunction. And third, a translational value. By bridging biopsy to therapy, we're closing the gap between clinical presentation and personalized treatment. And importantly, also, the system reduces reliance on animal models while retaining high physiological relevance, which we know is very relevant right now in the field in bioengineering and biomedical research. Looking ahead, my broader vision, I'm a postdoc right now, and I'm looking forward to move forward into my career. So my broader vision overall is to expand this platform toward other vascular diseases driven by mosaic mutations. I hope that by engineering vascular microphysiological systems, we can explore how these mutations alter vessel development, signaling, and repair, ultimately informing precision therapies for pediatric diseases. And this work has been a true team effort. I want to thank my mentors, Dr. Brandon Dixon and Rosanna Sanchez, my collaborators across Georgia Tech, Emory, Children's Healthcare of Atlanta, and even CHOP, and the many students and technicians who have contributed. I'm so grateful for the patients, families, and caretakers that have donated their tissues for these studies, and I'm very grateful for the Lymphatic Malformation Institute for their funding, NIH, and the Georgia CTSA program for supporting my research. And I'll just leave this here. I'll close with this thought, and it has my contact in there if anyone wants to email me about anything very specific. But by engineering human vascular tissues from patient-derived cells, we can uncover how somatic mosaicism can drive the endothelial dysfunction and hopefully bring precision medicine closer to every child that's affected by vascular and lymphatic anomalies. Thank you, and I'm happy to take questions.